So with that, I'll pass over to James to give his talk in 4 parts.
Thank you very much. It's really wonderful being here, and I'm really enjoying seeing this area of France. I had not seen before, and I've gotten to know people in it before. I am thinking about how I would structure this talk. I thought what I would do, and this is what the 4 parts basically are, is to give you a little bit of a background on what types of questions do we look at with the element sulfur. And I've, up to this point, since being a postdoc, before that I worked on oxygen, I worked almost exclusively on sulfur. I think moving forward, it's going to switch to methane and some other gases because of some instrumentation that we have recently acquired. But I will continue with the sulfur questions. So the first part is to sort of take a walk in from far away, things that we actually probably may or may not be able to measure, all the way through to things that we will be able to measure. So we're moving from far and deep space to the earth and to the oceans and to the rocks. And then I'm going to follow that with a brief introduction, brief description of isotope effects and what it is that we measure with the isotopes and what we're trying to sort out. And I'm going to focus on one type of isotope effect called a mass-independent isotope effect. And I'll give you an example from some work that we've done in collaboration with the group at Grenoble on ice core sulfate from stratospheric volcanic eruptions. And then I will follow with a 4th part, which will briefly introduce some of the background on using mass-independent fractionations to study what happened to the Earth through its evolution, its atmospheric evolution, and then a small experiment that we did last year, or that an undergraduate student did with me last year to test one of the pathways that addresses a question about how do you transfer some isotopic signature that's produced in the atmosphere that tells us something about the atmosphere to the rocks. And there were a variety of different hypotheses about this. Some are very specific, and we decided to go test one of them and see. I think it works. It works pretty well, but it brings biology into the system. What we're looking at here is an image of the Orion Nebula, and I'll see if I can switch it. Switch. Doesn't. Oh wait. Okay, I'm switching on my computer, but not on that computer. So let me figure out what is happening.
Yes, it's
okay. Did that go? Okay. Yeah. And so we have the Orion Nebula here. This region here is called the Kleinman Low region. And it has been studied spectroscopically. And when they look up through the telescopes and they measure the different isotopic and chemical species, they see things like methane thiol, they see things like sulfur dioxide, carbon disulfide, carbonyl sulfide, diazone. They're in a variety of different oxidation states. They also see ions, they see radicals, they see a whole variety of different species. And this, in principle, should give sulfur an extremely rich isotopic chemistry. It has a rich basic chemistry. They also see a 36 depletion. So there's a 36 anomaly. And there's another problem, which is that they don't see enough sulfur. Sulfur is supposed to be the 10th most abundant element in the solar system, and the amount of sulfur that they see is much less than that. So that's sort of a basic question. If we want to move forward in time and in space, we could think about how our solar system formed, and there's some questions that come up related to that. And this is one of the big bottlenecks for trapping sulfur isotopic signals that would be, say, of nebular origin, is that when the solar system went through this process, most of the sulfur ended up as iron sulfides, as troilite, in solid form. And that also accounts for some of the sulfur depletion in the Orion Nebula. That bottleneck effectively homogenized out, or is thought to have homogenized out most of the signatures that would have been produced. However, some signatures still exist, and this is a picture of the Murchison meteorite, which actually carries some unusual signatures in elemental sulfur compared to the sulfide sulfur. It tells us about chemistry that went on early in the solar system. It actually also tells us that at the time this thing was forming, the sun was illuminated because you had to have that sort of source of energy to produce the effects. The pictures of the people in the lower part of the diagram here, it's Daniel Eldridge, Abram Levidi, and Harry Oduro, were the people who had done this work. Now, you can move from this to building a planets, differentiated planets. If we look at iron meteorites, you see the iron nickel metal here. This is the Grant 3AB iron meteorite, and you see these troilite grains. And here we see there's immiscibility between the two, and there are signatures that tell us about how the planets were formed, related to the sulfur that is preserved in a variety of iron meteorites. You can also go to the silicate parts of the parent bodies, and you can study things like more primitive angrites, eucrites, and you can study the silicate parts of the Earth, and you will find that they do carry signatures, isotopic signatures, that say something about the way that sulfur was processed and the way that sulfur was introduced to the different parent bodies from which they formed. As well, there are ostensible signatures associated with metal silicate segregation and partitioning of sulfur isotopes between the two, and also signatures associated with sulfur loss, volatilization, melting and vaporization of sulfur. If we move to planets, this is a picture from the Spirit rover. It's false color. This is false color of sulfur on one of the rock abrasion tool. Sulfur is present on Mars. It actually has signatures that tell us about its chemistry in the Martian atmosphere and as well as the Martian surface. And we'll look at that. Some of the information that we have about its chemistry come from studying Martian meteorites. These are three of them. This is Nakla, that's Shogati, that's Lafayette. This is an titanomagnetite inclusion. Unfortunately, you can't tell the brightness, but some of these are slightly brighter, and are sulfide minerals that were formed in a Martian meteor, a Naklite Martian meteorite. It's elephant moraine of 3346. And this tells us something about the compositions of these sulfur, tells us something about where that sulfur came from, and is actually also intimately connected to the oxidation reduction chemistry that produces titanomagnetites on emplacement and on the cooling of this meteor. These are some of the types of questions that we look at and that other people look at, trying to sort things out with isotopes. We also turn our focus, and I'm going to spend a lot of our focus, into sulfur chemistry that occurs on Earth. And so in terrestrial environments, we have rich low temperature chemistry and rich high temperature chemistry. We also have chemistry that occurs in the atmosphere. And all of these things leave behind signatures that can be studied. In the oceans, sulfur is the most abundant, or the second most abundant anion is sulfate. And the reduction of that sulfate to iron sulfide accounts for about, I think it's about 50% of the remineralization of, the remineralization of organic matter in sediments. is a very significant part in it. It also accounts for about 30%, the other 70% being made up by burial of organic carbon of the net oxygen production long-term by our biosphere. So the formation of pyrite, reoxidation, and then ultimately the burial of carbon. So it's a system that we want to know about to understand the environment today and in the past, and how to connect these geochemical cycles. It is connected through a variety. This is a figure that's taken from a paper by Jakob Zagfi. It is connected through a variety of reactions that relate sulfur of different oxidation state. And sulfur being bonded to two hydrogens is in a very different bonding environment than sulfur that's bound to four oxygens and sulfate, or to any of these other intermediates. And what these different bonding environments mean is that for isotopic chemistry, which we'll get to, the magnitude of the isotope effects that are seen and can be measured end up being very large, because there are, as you change the number of bonds, as you change the strength of the bonds, you change the way that isotopes are partitioned. And you can find evidence for these transformations. This is in carbonate bars in Western Australia. I think that is my son. And there's a lot of sulfate that's in the water here. If you look down and you dig down in the sediments, you'll find some dark areas. And you dig down, it's a grayish color. It also has this characteristic smell of sulfide gas, which is not stable at Earth's surface, but is produced by organisms that are using sulfate as an electron acceptor to consume organic matter and to live. If you look close, this is near where I live on the Severn River. If you look back in this part of the river, there's a large bay, and there's actually there's a sill that runs across there that effectively isolates that bay seasonally. Now with more people living in this area, more nutrients go in, and now this bay goes seasonally anoxic, which kills off plants, kills off fish, and is of interest. It also turns the sediments in the bottom of that bay a really deep blue, black color. They also have this characteristic smell of sulfide. And in addition to forming that sulfide, in addition to forming those blue-black, which are iron sulfides, some of those sulfides end up making it down, being recycled into the deeper parts of the earth. So this is the Europa kimberlite pipe. These are sulfide inclusions in diamonds. This is, I think, from Emily Tomaso. This is one that we studied. Those sulfide inclusions carry isotopic signals that tell us that they were at the surface at one time in Earth history, and importantly, they were at the surface early in Earth history. And so they were processed through the atmosphere and somehow made it into the source regions of those diamonds. I'm not going to talk about those, but Emily has a whole bunch of work on this, and it's an interesting question about how you get them there, how you keep the sulfur intact and from mixing with other sulfur that would be in those source regions. This is a picture of Ofu. It's I can't pronounce it. The other island. It's in Samoa, and here there is evidence that in Samoa and then also in Ofu and some of the other islands in the Cook Islands, there's evidence for recycling of sulfur into the source area for the magmas that formed these islands. Some of that may be very old. Some of it appears to be clearly younger. The work is done by Gibran Labidi, by Matt Jackson, and others. In OFU, there's an unusual or strange thing that we're trying to track down, which is an apparent correlation between sulfur isotopic compositions and tungsten 182, a highly siderophile element that tells us something about potentially very early and ancient reservoirs in the mantle. And the question is, and that we're trying to work out, is trying to understand what is the nature of those connections. What does that tell us about the sulfur? Are we dealing with sulfur that was well mixed in the earth or not? It's primordial. Are we dealing with the recycled components or not? And how do we understand that? So those are the sorts of problems that we tackle. The way that we have tackled it is using a variety of different approaches. We take samples, we analyze them, we have experiments, we analyze the products of the experiments, and we try to understand or capture the signatures of chemical and physical processes that change the isotopic composition. Now there are a number of reasons why isotopes may fractionate. I'm going to go through two basic classes and some of the notation related to those. So for sulfur, we have four stable sulfur isotopes. We have sulfur 32, 33, 34, and 36. If you're working in the atmosphere today, you could also look at sulfur 35. It's an 80-day half-life isotope and used for atmospheric questions in addition to these. The compositions of the isotopic compositions are generally given using this delta notation, and I'll use that a lot. Basically, that notation is really just the deviation in the proportion, say, of 34 to 32 in some sample relative to a reference material, and then it's expressed in parts per thousand. The -1 is the deviation. In addition to that, and because we can separate out different types of processes, there's a second notation that we will use. And that one, instead of being a lowercase delta, is an uppercase capital delta. And this one is simply taking what would be effectively this delta value for, in this case, it'd be 33, 32, and then subtracting, taking the deviation of that from a predicted value from the measured 34, 32. So there's a correlation, and I'll show you in a moment, between, say, delta 34 and delta 33 that is well behaved, and we understand the reasons for that. And then in some cases, the rules are broken. We also understand some of the reasons for that. When the rules are broken, this quantity comes out as non-zero, and when the rules are followed, this quantity is pretty close to zero. It's not always zero because there are variations. So the question is, what are the rules? What are the things that control that relationship where this becomes zero or non-zero? And for this, I'm going to go into a little bit of background on isotopic fractionations, on the fundamental basis for isotopic fractionations. And you may have heard there are equilibrium effects when two substances or many substances are allowed to effectively reach their energy minimum state, and then they partition elements and isotopes between the different different substances in certain proportions that depend upon their affinity for the different materials. For isotopic substances, when we're looking at an equilibrium fractionation, and this side is the equilibrium side, you may think about it in terms, say, of two bonds. Okay, there will be more than two bonds, and those two bonds will have a certain potential energy surface. Say this one is a tighter, deeper, stronger bond. This one is a weaker, bond here, slightly broader potential energy surface. And if you partition energy into those substances, the atoms have to move. Okay? And they move at different rates depending upon their masses. They basically sit, and that means that they sit at different energy levels in the potential well. So the heavy isotope in this bond will sit here. The heavy isotope in this bond will sit here. The light isotope in this bond sits here, and the light isotope sits here. And basically, the partitioning is a competition between those energies and the drive to randomize the system. And this thing depends on the way the atoms move, and it depends on the masses of the isotopes. Likewise, you may have something that people talk about as being kinetic. It'd be a unidirectional process. And typically, people, when they talk about something like evaporation or something like that, they just talk about it in terms of 1/2 mv squared. So they talk about this thing is escaping and it's moving at a specific rate. In reality, when you're breaking a bond or you're evaporating something, there's a whole chemical potential energy surface that it has to follow. And so if you're breaking the bond and it's a complex material, it will go up through some sort of potential energy hump, and then it will escape. The same thing can happen. This is in cis and trans ethene, so it's just a schematic. What happens between two doubly bonded carbons with hydrogens and say chlorides here and there? They may have slightly different energies associated with them. You can calculate this. If you want to go from cis to trans, each of them will have its own potential energy well. This will be some other coordinate that's related to that. How this and this end up flipping, because this is a double bond, so it can't just rotate. So it has to go over this energy hurdle or threshold here, and at that threshold, there are movements that can exist in orthogonal directions. Okay. Those things are also vibrational. This traversing of this hump is essentially, it's the inverse of a vibration. And this difference allows for, if you have this heavy isotope here, light isotope here, it will populate heavy isotope here, light isotope here, and there will be exchange that goes back and forth, and then a certain amount will react across depending upon how much is in this state. And that actually determines the rate at which each isotope reacts in the reaction. And so you will actually have the light isotopes will react faster in this case, because this difference and this difference are slightly different than the heavy isotopes. And you will get an isotope effect that's a kinetic. As soon as these two populate, you get the forward and backward, and you can minimize the free energy. Both of these things if we looked at delta 33 versus delta 34, depend on the mass of the isotopes being substituted. For 33 versus 32, which is this ratio, it's 1 atomic mass unit. For 34 versus 32, which is this ratio, it's 2 atomic mass units. Basically, ends up with a very tightly constrained relationship for those types of fractionations that generally follows a relationship where the sulfur 33 is fractionated by 1, whatever unit per mil or whatever, and the 34 is fractionated by twice, which gives you this really tight, and this is real data, just measured randomly around the world for samples that are younger than about 2 billion years old. Older than that, things sort of fall apart. I'll explain that more later. And this just arises because of the role of mass in translations, rotations, vibrations in the materials. It also does not produce any deviation from this line, which is the reference line, so that capital delta is 0, or essentially 0. Now, if you have a photochemical process, or a process involving a radical, or an ion in gas phase, and there are other processes involving, say, electronic spin, coupling between electronic spins and nuclear spins. you can have other types of selection factors that enter into the isotopic selection. So what I'm going to do is I'm going to say, this is a diagram. It's actually from a paper by Shuhei Ono that describes the background of mass-independent fractionations. This is a diagram that shows the potential well schematically for SO2 in the coordinate related to the SO2-SO bond. Okay. And then this is the ground state. And you have a given vibrational level here. He's only drawn it for one isotope. And then in ground state, the probability of finding the bond length is highest. It's normal bond length, then trails off as it goes side to side. If you absorb a photon of light, you kick it up in terms of energy, and you will effectively change the angle of that SO2 molecule. You'll change the bond length a little bit. You'll change the strength of that. Because what happens is a photon comes in and it kicks an electron that's in one orbital out to a slightly further orbital out. Okay, so it becomes a different substance. That substance can also have vibrational energy levels. But since you're coupling between a photon, it's going to couple with whatever vibrational energy level it can hit. So you will have preferences when you hit each of those vibrational energy levels. And then that thing may oscillate around, and it may hit a surface like this, which will dissociate it into SO plus O, and all of a sudden you've got different sort of chemistry. Each of these steps, the absorption of light, the coupling between these states, even the coupling in this state itself, are complex. Recently there's some work with this particular state that, when I first saw it, I thought of it like a lake with the energy rattling around inside some sort of nicely shaped lake. It turns out that energy surface is shaped more like a dumbbell. And so there's a restriction in the middle to get from one side to the other of this potential well in this third dimension. Any of those steps don't strictly depend on mass. And so those steps then produce a different type of effect that deviates from this line. And this is what I ended up, I was actually looking initially for mass-dependent processes. I ended up finding things related to mass-independent processes in the geologic record, and that changed the focus of a lot of the work that I ended up doing for a large part of my career up to this point. Now what I want to do is, these are Archean samples, these are Proterozoic samples in blue. Now what I want to do is I want to, I'll just check the time, okay, good. Is I want to take you through two examples of situations where, and examples of how we use these signatures of mass-independent chemistry to study processes. The first of these, first example is going to be more recent, last 2,000, 3,000 years. And this is the work with the group from Grenoble. And then we'll return to this, and we'll return to some questions related to it. If people have questions, you should stop me and ask. But we'll take it from there. Okay, so when I was at UCSD as a postdoc, Joel Savarino was a postdoc there now. Now he's at Grenoble. He originally discovered much of what I'm going to talk about. And then he had a student, Melanie Baroni, who discovered some other stuff. And one of his recent students, Elsie Gauthier, is writing up some papers on some work that they have done, which has, I don't know, probably taken about 10 years to be able to do, because they had to go to Antarctica, drill a number of cores, characterize all the cores, cross-match them, cross-correlate them, and then extract the samples. And then they came to my lab to do some of the analyses, then some more easier techniques were developed, so they've moved on to doing that. And what their interest was, and still is in trying to construct a chronology or a history of stratospheric eruptions. going back in time, and it's for climate purposes. The basic idea is this, and we'll look at the parts in sequence. If you have an eruption that occurs, and it's very large, it can send gases into the stratosphere. Very quickly, those gases undergo atmospheric reactions, but often involving light, and they convert sulfur dioxide to sulfuric acid aerosols. These things in the stratosphere, they don't sediment out. In the troposphere, they do. And then if they're near the equator, they can cross from one hemisphere to the other, and they can then be transported poleward by a circulation known as Brewer-Dobson circulation. And then on the poles, they sediment out everywhere, but on the poles they sediment out and they are trapped in the ice as layers that are sedimented out over a period of years. And the question that comes up is, if you have a volcanic layer, is it a local volcano or is it a stratospheric volcano? Does it have a climate impact? And if you have a chronology which has been done and is very valuable, that is based on correlation of volcanic horizons in the northern hemisphere and the southern hemisphere, can you actually know whether you had two local volcanoes or one big equatorial volcano? Or can you know, if you only have a deposit say on Greenland ice, and you're trying to match them side to side, can you know whether that one was a stratospheric volcano that just didn't happen to cross the equator? If you have a hemispheric effect, it still could have a significant climate effect that we want to understand. And that is what these two are and were focusing. This is some of the data. This is the concentration. These curves are the capital delta 36 and capital delta 33 for a series of horizons through the I think it's the Samalas eruption, which I believe is in Indonesia, that happened about this time. I think that the dates are because I think 1257 they see a signal showing up in the ice core age 1256. What is seen is there's this characteristic spike in concentration. That's the sulfate that's being deposited over background, but it includes the background signal. And then the 33 signal is near zero, and then it rises, and then it drops. So it rises to positive values, drops to negative values, and then it recovers. And likewise, the 36 signal does the complement of that. That characteristic shape is what's seen in other eruptions. These are on the same time scale, a variety of other eruptions that they have studied. That's the characteristic signal of this stratospheric chemistry that allows them to use these as markers of reconstructed records. Now, there are some things about this as we worked through this that I found really kind of interesting, and I thought are worth pointing out and worth talking about. And I wanted to go through that in more detail and look a little bit more at the isotopic records for this eruption, and then look at or consider some of the processes that occur in the path from volcano to stratosphere to ice. This is a picture, I think it's a shuttle image. It's from NASA, taken about 2 months after Mount Pinatubo. There are some dark bands in here that are sulfuric acid aerosol bands from that eruption. These is effectively the sulfur dioxide is transformed into sulfuric acid, and then it gets mixed fairly rapidly horizontally throughout the stratosphere. Now, what's puzzling about that, and this has been noted, is that the timeline from 1256 to 1264 is not 2 months for this depositional signal. It's something that is taking, effectively, it looks like about 8 years. So they're producing some sort of signal in the atmosphere very, very quickly, but then for some reason it ends up being preserved in the atmosphere, potentially altitudinally, if that's the present working hypothesis, and then it sediments out with time, preserving that without remixing everything back down. So there's something going on there that we don't understand, but that is a question to understand. The other thing that's interesting is you can plot this is just the capital delta 33, this is the capital delta 36. You can plot the capital delta 33 versus delta 34. The same thing except the inverse for the capital delta 36. These points are numbered. This is the first time point, second, third, fourth, fifth, sixth, seventh, eighth. Okay, this type of sequence was observed by Melanie Baroni and is observed here over and over again. And it tells us something about what is it, how is the signal being produced, and what is happening. So this is the background sulfate isotopic composition that's being deposited. And then it very quickly, these are background corrected values, not based on trying to fit this line. The line just goes there. These are based upon, say, taking chloride to sulfate to know how much additional sulfate they had in there versus how much was from ambient signals. And basically, what happened was you mixed in some component that itself was evolving with time. Okay, and part of that evolution is a distillation. It is interpreted as a distillation signal. So you start at one composition, and then as you run the reaction, you drive the residue away from the product, and the product then essentially follows the residue. This tells us something about the chemical reactions that are in this reaction. And we can go back and we can say, okay, is the chemistry consistent? And that this is what they did from volcanic eruption to volcanic eruption. And so far, with, and these are the corrected values for a variety of different eruptions for the capital delta 36 versus capital delta 33. There's the ones in color. The gray boxes are ones from the Earth's early geologic record. And so far it appears that there is this consistent relationship from eruption to eruption and actually from the Archean to the present.
So just to be clear that you did the same calculation for 36 for this mass balance calculation, and then you plot.
Yes.
Okay. And for the each single point.
And to some extent, this tells us, okay, that looking at this record should be a good way to know if we have a stratospheric eruption. There's some other levels to this that Joel has looked at, which have to do with the O17, which I won't talk about. But he sees signals in O17 that appear to be connected with the largest eruptions in time. So there may be something there related to the chemistry that shows up in the oxygen isotopes. And so, so basically, they did this for a whole variety of eruptions. They have got their record of stratospheric eruptions, and then they also have records they can compare with, which are the state of the art, where they compare chronologies in the northern hemisphere with the southern hemisphere to say which are bipolar eruptions. And the inference is that if you have a bipolar eruption, a lot of sulfate coming out is probably a stratospheric event, and probably not something else. However, you do have the potential for getting two local eruptions essentially simultaneously to make it complicated. or the potential for a high magnitude eruption. And this is the match between these two, and basically the key here is that these eruptions here that were thought to be single pole eruptions do carry a stratospheric signal. So there are some eruptions in the record, and then some eruptions, if you go very old in the record, that don't match. But generally, the matchup between the methods is really quite good. And so what Joel and what I think Elsa would argue would be that this is not a replacement for the standard methods that are used, but it's another tool that can be used to get additional information and be able to understand the eruptions that occurred in the past, and then potentially be useful for climate studies. Okay, now I'm going to move back in time to some of the early stuff, and this is moving back to well, basically throughout Earth's history. This is a picture of the Hammersley, the banded iron formations from about 2.5 billion years ago. And when I was a postdoc, I was measuring samples specifically to look for a biological signature. So I actually expected to see something very much like what's on this blue line relative to this capital delta. Some of these blue line points don't match exactly with the zero line, but those points can be matched up to the metabolic effects that can be resolved. And if you have this in combination with the 34, you can provide a consistency argument for studying metabolism. And I was looking for those same signals back about 3.5 billion years ago in some samples that had been studied before. And I did some measurements. I'm assuming they're probably somewhere in this range. And they didn't fall on the line, and they were way too big to be biological effects. And so first thing I did was I called my wife and said, okay, something's gone wrong, I'm coming home, I'll come back to this tomorrow. But then I went back the next day, we redid the analyses, or I redid the analyses, I did more analyses, and saw this signal. And I was in a lab that was studying the atmospheric chemistry questions, so that was the first order interpretation. I wasn't actually the first person to see this. Some other people had seen it, but they didn't have the context to make that interpretation. Harold Strauss, I think, worried about his mass spectrometry when he was doing his habilitation in the 1980s, and Tom Herring published some results that he said, well, maybe the Earth isn't like what we thought it was like. But I think they were probably scratching their heads afterwards. Basically, what we see is we see a period early in Earth history where there are very strong positive and negative mass-independent signals. Okay, tells us something about the atmosphere. It's analogous to the things that we see in the ice cores today, but it's something that would have had to have been much larger scale because we don't see the signature trapped in the rocks today. And so the questions that come up are, what is the reason for this, and then how and why is it preserved in the rock record? And the reason for this takes us back to this diagram, this photochemical diagram, and the chemistry that is required to have the radiation that's required at that sort of, these sorts of energies apply and make these sorts of banded absorption spectra. And it's a large amount of energy, and the amount of energy increases as the wavelengths get shorter. And so the region where this chemistry occurs is really below about 325, below about 230 nanometers in terms of the wavelength of light. That's very deep UV. We run into our UVB in this region, and we're shielded because of ozone, which exists because of oxygen. The only place where you don't have it is when you get up to the stratosphere. When we're up there, we see the same signals. This banding here is just a result of this coupling between those different bands. And one of the key points here is that the heights of these peaks, these are for two different isotopes, actually differ. And they don't differ in a mass-dependent way. And that's part of the reason for the effect. There are many reasons for this. But the implication is that we have something that is a very short wavelength. There's another hypothesis that says it's not linked to SO2, it's linked to sulfur recombination chemistry. But again, you need to make the elemental sulfur, so you still need the radiation. So just to give you an idea of how it connects, this is an older slide, maybe some of you have seen this. This is the intensity of, this is a calculation I did long ago, of the radiation watts per meter squared per nanometer of radiation. This is what's coming in at the top of the atmosphere. This is the radiation that we see today. If you do the calculation, it's like latitude of, I think it's about 50 degrees north. And then if you drop the oxygen level by a factor of 10, you see it starts to increase, and then you get a factor of 100. You start to open up a window in here, a factor of 1,000. You've opened up a much larger window. This is actually absorption by ozone. This is absorption by oxygen. And then you go to 10,000 and 100,000, and effectively it's transparent. So to turn on and off that chemistry does require something that allows for the atmosphere to be transparent, and the biggest connection is oxygen. That's one of the connections that was made in making a link to oxygen. There also was another link that was made very quickly by Jim Casting and Alex Pavlov, and they described that connection using a diagram like this. Now this has your two volcanoes in it, one introducing H2S, one introducing SO2. On the bottom axis is the oxidation state of sulfur. And they said you can have a reaction where it absorbs light, HNu, to make HS. You could also have it react with HCO to make H2S. Or you could have this HS react with an S to make an S2, or with an HS to make another S. And you can connect all of these reactions up. In today's atmosphere, reactions like this occur. And there's enough oxygen so that effectively anything that goes this way gets funneled this way, and everything leaves the atmosphere as sulfate. And so you don't see these signals show up except for if they're separated in time, which is what they are in the ice cores. So you don't get them preserved in the rock record. If you go back to a time when you had low oxygen, so these two channels become less intense, you can then have S2 reacting with S2, S4 reacting with S4 to form S8, etc. And all of a sudden, you can have sulfur leaving the atmosphere in two forms. Now, their original idea was it's two forms. It could be more, but the key is that you have to have it leaving probably by two pathways, and that's the key. For this one, they come up with the threshold about a million ppm oxygen compared to today. So the idea is that throughout the atmosphere, oxygen was the controller of the. Let me check this. Of whether the signal appeared or not. And that basically implied that this was the time at which oxygen rose to significant levels. You could have had oxygen that increased before that, but not sufficient to have completely turned the system to more of an oxic environment. Now there are, I guess I thought I had a slide in between the two, but the question then comes up as to why then, in the geologic record, do we have a record with both positive and negative, capital delta 33, in one mineral, effectively in sulfides and pyrite, which are preserved? You do have sulfates that sit down here, but the question is, how do you get both positive and negative signatures into the rock record? And this is a question that has been, ever since this discovery was first made, and has continued on through to the present. And this is what I want to sort of wrap up with, or want to wrap up with. The first thing I want to say is generally we need biology, or it's thought that we need biology to make the pyrite, to make the sulfides that are going to be preserved in the rock record. And so we need a very ancient metabolism and an ancient way to produce the sulfides. And the evidence, there are a variety of lines of evidence, but the evidence from, say, genomic treatments suggests that the metabolisms that, that are very old for sulfur reduction, for sulfate reduction, for SO3 reduction, evolved very early in our history. And the isotopic evidence also suggests that they evolved very early. There are connections, sulfides in barytes by Shen et al. There's this Potognali study with carbon and sulfur. There's some very large fractionations in organic associated sulfate, and there are some relationships that suggest that sulfate reduction was something that operated early. But the question still remains as to how do you make the link to something other than the sulfate pool? How do you make it to the elemental sulfur pool? Because these things all have to do with sulfate reduction. They don't have to do with elemental sulfur. And Shuhei noticed this, and this is in one of his early papers. He argued, he said, well, maybe seawater sulfate was here. Maybe sulfate reduction produced some of these signals, and maybe there was an atmospheric signal here. And then somehow you fill in that region. But you've got this triangular shaped field that develops, and we want to explain it, and we want to explain how that occurred. Some of his attempts had it where you have elemental sulfur or gas exchange between H2S or H2SO4, and then once it gets into the oceans, you get cycling, and he invokes a species, an FeSH species. He invokes potential ways of getting S naught or S2O3 into pyrite, but the real question is, how does that occur? And one of the breaks, or one of the ways that this might occur, comes from a proposal by George Luther, and he also proposed something similar to this later as a potential explanation, where here's this FeSH, but it's reacting with the polysulfide here. And basically this reaction takes this FeSH, drops the SH here, and cleaves two of the sulfurs from this polysulfide. Okay? And then that makes a precursor for forming the pyrite. If this polysulfide can then be linked to the elemental sulfur, maybe that's part of the key. And this is one way that it might work, sort of a schematic. You have seawater sulfate here. It goes into the sediment, becomes pore water sulfate. You may have reduction to form sulfide, which will form pore water sulfide. That's the stuff that we dug up. It's made the sediment smell. And then the sulfide may go to form the pyrite that is formed. Likewise, if we look at the elemental sulfur is insoluble, end up in the sediments, but then it can react with and exchange with the sulfide. If it reacts, it forms the polysulfides, and that too can go to pyrite formation. Or if it exchanges with the sulfide, one sulfide with an S8 will form a whole bunch of polysulfur. This sulfide can then, in fact, transfer the signal to the pyrite. And this is where this is sort of the question that we had, and this is where the work that came up with Liz Peters. And so for her senior thesis, she grew a series of cultures. These are a set of 4 different experiments in batches of 3, which we learned today was important. One, there's an abiotic control. There's a control with bacteria plus elemental sulfur. There's a control with bacteria plus Fe2 plus in solution, and then there's an experiment with Fe2 plus and elemental sulfur in bacteria, which is what we would postulate would be relevant. In the one without the Fe2 plus, you form this nice color. This is polysulfide. So the sulfide reacts with the elemental sulfur and pulls it into solution, forming the polysulfide. In the one with the iron, you form a lot of iron sulfide precipitate, form even more when you have the 3. And basically, just to show you quickly what the results were, and the way that she did this was to start with a sulfate that had an isotopic label that was mass independent, and use normal elemental sulfur. So actually, the sign is flipped from the way that we think it should occur. Her starting composition was here. This is the results of an experiment with just iron and bacteria and sulfide. The residual sulfate ended up being enriched, so you remove some of this light stuff, you push the residual sulfate this way. The injection of the inoculum, so the bacteria actually dilutes this signal and shifts it down this way. And then we form FES and FES2 with a small difference in composition. And this is sort of what we would expect. They're all mass dependent. In the experiments with the elemental sulfur present, she saw something very different. And so with the one that had elemental sulfur and no iron, we don't form any iron sulfides, and so basically we get this composition. The elemental sulfur buffers the system for the products, and the sulfate is enriched. But we are somehow, because this is pushed this way, we must be producing something that is negative. So there's something going on. With the ones with iron and elemental sulfur, we ended up with the FES here, the FES2 here, and the elemental sulfur down there. And if we take this diagram, this is sort of the opposite of what Shuhei's diagram was, but I took the liberty of using Excel and dragging it and switching it upside down. If we do that, this effectively is the same thing as what he has. And I think we're getting closer, but I think we have a viable way to explain why the signature is both plus and minus in the geologic record. And we have and we can now start trying to take apart the geological record to understand what the conditions are that cause the variations that we see. So what I hope I've been able to do today is to show you a little bit about how the fractionations occur, show you a little bit about how you can use them in modern systems to get information that we think is useful for people nowadays trying to understand what is the role of volcanoes in climate, some explanation of the deep time record, and then a hypothesis and ongoing work that we're doing to try to understand how to reproduce that. So I'll stop there. Thank you.
We have questions.
Yeah, why do you think there's no correlation between the total sulfur in the ice record and capital delta fractionation? Well, if you look at the histogram there,
Yes.
you have the highest total sulfur emitted. You have no fractionation.
Very, very
Yeah.
little. Yeah, at the crossover.
So the two signals must be of different origin.
I understand your question. You're saying that if
you have no very little sulfate or
Right.
total sulfur emitted, you got no fractionation, and if you have a lot,
it comes back,
Yes.
is what you're saying,
Yeah.
yes. So I can tell you our bias in interpreting the record, and then we can go from there. Our bias is that this is a time-dependent signal that sort of is a pulse that is fractionated and we're capturing that through time.
sorry.
I'm trying to think about a distillation curve, and
So could it mean that the largest sulfur emitting eruptions don't reach the stratosphere? I mean,
Oh, no, I don't. I think for the signal, I think it has, we don't see the signal in the troposphere. So we're pretty, I mean, the community is pretty certain that it is a stratospheric signal. And this is a single eruption. I think the question is how this transposes, and I know we talked about this, but I did not go through the calculation. I asked about it. How when you take a distillation curve, when is there?
This is John.
Yeah. So. If amount, I can do this. So if we went if we looked at sort of a Rayleigh type effect, you would expect to start off with a curve something like this, with this difference here being the fractionation for that reaction, or being associated with the isotope effect for that reaction. And this thing will, the reactant will, as it's consumed, will drop, and the product will chase it. And so the question is, if we're reintegrating this curve into pieces like this, where we take, okay, this is so much, this is so much, this is so much, do we end up with the middle parts, which are most of the reaction, averaging out to a value that's near 0 or no? Now, why I can't answer that question directly is because when I do know, in spite of not having done this, but from the questions that we asked, that when we look, if we try to reintegrate this curve, we appear to be losing earlier, or we appear to lose, I don't know whether we lose them. We're not able to reintegrate it to a value of 0. So sulfate is not a full representation of the concentration or amount versus the fractionation of the product is preserved in the ice. There's some loss somewhere. But this is that's the model and the bias that we have, and I think it probably is something along the lines of it's the central part.
So do you think the isotopic composition of the stratosphere is homogeneous throughout the globe, or if there's some kind of
no, well, after an eruption,
yeah.
I think the dynamics that we were reading and trying to understand what it is that would distribute the aerosols, and you have to separate the aerosols from the reactant, from the SO2 during the process, which means that it's not simply entrainment or there's some level either of settling of the aerosols relative to the SO2 if it's rising due to thermal buoyancy forces or descending.
I was also thinking about some like strength is not necessarily the same throughout the.
It's not the same, and the other thing is that aerosols will migrate if heated on one side. Some of them will, so you can actually separate them using a photospheric effect. So that and so we think that these different parts of this signal may be capturing different altitudes in in the stratosphere, where those aerosols ultimately reside. What we don't know is whether the ones at high altitude are first formed or last formed, whether the ones here are first formed or last formed. We don't, those are questions we have not, we don't have good answers for.
There's a question here too.
Yes?
Do you have Cap 36 anomalies in the Archean?
We do.
Correlated in the same way as found in the...
So they are, the capital delta 36 anomalies in the Archean show a large number that appear to be very similar to the ones in the ice core. There are some notable exceptions where they steepen, and there may be some where they shallow a little bit. I'm not aware. But the relationship between the Cap 36 and the Cap 33 in the Archean varies, and it appears to vary in a systematic way, where if you measure individual layers in a drill core, for instance, you'll find excursions in the relationship between 36 and 33 that correlate with other things. And part of the interpretation of that is that that's due to a change in shielding that's occurring in the Archean, potentially related to methane or something else that's changing either the light or the reaction networks that apply. So there are signals, but it gets more complicated as we look deeper.
Last call for questions.
I'm amazed that your experiment on sulfur isotope fractionation with the bacteria,
Yes?
you have the same amplitude of fractionation as Shuhei did? Is that pure
It's just luck.
luck?
No, we
Okay.
bought, we wanted, so I use methods that take a long time to do analyses and are really painful. And for an undergraduate project, we couldn't do that. So we needed a signal that was big enough to see that did not swamp out my colleague's mass spectrometer. And so we bought some 33, we did a calculation, and we got a value that was about 10 to 20 per mil. And we mixed it up, it was 14. And that was how we did it. Yeah.
James will be around for a few more months yet. If you want to question him further in the hallways down the weeks, he'll be here. I'd like to thank James one more time for other people.
Thanks to you too.